Warm greetings, everyone. I'm Ajay Vikram from India, and I'm here to give a talk on the research titled as Comparative Analysis of Evolution of DNA Gyrase. It is a study on the phylogenetic analysis of Halobacterium species NRC1. In the evolutionary tree of life, there exist three main domains of life, namely bacteria, eukarya, and archaea. Whereas bacteria and eukarya are found in the normal conditions of Earth all around the environment, archaea, which is also termed as the extremophiles, are found only at the extreme conditions such as high salinity environment, high temperature, high pressure, high pH. So in such harsh environments only, these type of species thrive. So studying more about these species, evaluating their genome and arriving at a conclusion will help us or will provide us few insights on how species can evolve on such harsh environments around our solar system. So this is the basic idea of this research also. So Halobacterium species NRC1, it is considered as a good model or a perfect model species to evaluate for such a research. And Halobacterium species thrives only at high salinity environments. Even if they are kept at the low salinity environment, their proteins will not function because they have modified themselves to survive better at the high salinity environment. They grow on amino acids in their aerobic conditions, and their cell walls are quite different when compared to the normal bacteria and eukaryotes. Their shape can either be rods or cocci, and in color, it can either be red or purple. Halobacterium grows best at 42 degrees Celsius. So, Halobacterium species are commonly found in Earth in such places such as Great Salt Lake, the Dead Sea, or the Lake Magadi, or the Owens Lake. In such places, Halobacterium species are found predominantly. So the DNA gyrase is actually a type of type 2 topoisomerase and was found in the year 1976. So it is one of the type of topoisomerases which has the ability to manipulate the topology of DNA. It is comprised of two main subunits, gyre A and gyre B. And the final product will be of the A2B2 tetramer. The main function of DNA gyrase is to introduce the supercoiling in the DNA, and it introduces two supercoiling, negative supercoils at the expense of two ATP. And this DNA gyrase complex has three gates, namely C gate, DNA gate, and N gate. So during the start of my research, the main hypothesis was that as DNA gyrase is a significant type of topoisomerase, which is found predominantly in archaea and bacteria, so there might be some environmentally infused factor that led to the evolution of the DNA gyrase to work so efficiently, so perfectly in the Halobacterium species. And in order to study that, the idea was to compare the evolution of the protein sequence of the Halobacterium NRC1 with other extremophiles, then to the bacteria and eukaryotes. So I use various type of websites and tools for it, and I have given the basic idea of what I have done in the methodology part. I set up a local database of the 9 halos, and using branch orderers, I was able to download the whole protein sequence. Using Cluster Omega, I was able to get the protein, the percentage identity matrices, and using NG phylogeny, I was able to arrive at a conclusion about the evolution of the tree. And I use the 9 halos for this. And as of now, the first round of analysis was done, and it was so great. And I was able to arrive at a good conclusion. And the second round of analysis, at the end, I would be able to arrive at a more firm conclusion about the evolution of the DNA gyrase. So I take this time to thank my professor, Dr. Shiladitya Dasarma and Priya Dasarma, for helping me throughout the research. And I would like to extend my gratitude to my fellow colleagues as well. Thank you, guys.